Hey everybody, welcome back. We are going to start Unit 6 today. This is the lecture video for Unit 6, Lesson 1, Exploration 1. Throughout Unit 6, we're going to be looking at DNA. We're going to be studying its history, what it's shaped like, how it reproduces, and how it's used. And so let's go ahead and get right into it. Today we're going to look at the function of DNA. DNA is a really special molecule. It holds all of the genetic material for an organism. And we describe that genetic material as a set of traits. Okay, your traits are any of your physical characteristics that have been passed on to you from your parents. Anything that you have that makes you look a certain way that you got from your parents is a trait. So, for example, if you have like a nose ring, that's not a trait because you didn't inherit that from your parents. But the way your nose is shaped would be a physical trait. So when we talk about that, it's going to be important that we keep track of certain terms like a trait, because we're going to bounce back and forth between descriptions of different things. But whenever we talk about a trait, we are talking about one of those inherited physical characteristics. Now, as we've talked about before, DNA stands for deoxyribonucleic acid, and it is what you inherit from your parents. It is what all cells contain, like we talked about in the last unit. And even though portions of the DNA may not be used, every cell has every aspect of your DNA. But scientists haven't always known this. They've known that traits were heritable, meaning they got them from their parents, but they didn't exactly know that DNA was the molecule responsible for that inheritance process. And so today, I want to look at, how we kind of figured that out. All right, now let's go ahead and skip down to the three scientists, or the three groups of scientists, I should say, that are credited with discovering that DNA is in fact the molecule that contains the genetic material. I've highlighted this word genetics. We'll talk more about genetics as it relates to heredity in our next unit. And so some of the names that you'll see, like Gregor Mendel, we're not going to talk a lot about him right now, but we will discuss him in greater detail in our next unit. But the first person that I want to focus on is Frederick Griffith. Now, as I said, there's going to be three groups of scientists that you're going to want to be very familiar with. So as you take notes over this unit, make sure that you have very detailed descriptions, not only of these people, but also of the experiments that they did to contribute to the discovery that DNA contains the genetic information. All right, so let's start out. And the timeline here is important because that helps you realize, what came first, second, and third. So 1928 is our first scientist. It was a scientist named Frederick Griffith. And in his experiment, he decided to see, all right, can I point to the existence of some sort of genetic material? Because we were fairly sure it existed, but we wanted Frederick Griffith was the first one to get some data to that. So let me kind of describe his experiment to you. All right. He took two types of bacteria. He knew that one of the types of bacteria was going to be totally harmless, and that another type of bacteria was going to be lethal. And so I'm going to use this diagram to kind of show you what happens in this experiment. There's a description of it in this paragraph as well. But what he did was he took the S strain of bacteria, the S stands for smooth, and he injected that into the mouse. And when that happened, the mouse was totally fine. Then he took another strand of bacteria, the R strain. Actually, I might have messed that up. The S bacteria killed the mouse. Sorry. Then he took the R strain of bacteria, which was the rough strain, and he injected it into the mouse, and the mouse was totally fine. The mouse lived. Then he boiled the mixture that contained the S bacteria, effectively killing it. He injected it into the mouse, and the mouse was totally fine. And this was where so all of these three things were expected. He expected that when he injected the deadly bacteria into the mouse was going to die. When he injected the non-deadly bacteria, the mouse was going to be fine. And he even expected that when he boiled or heat killed the S bacteria and then injected it, that the mouse would be totally fine. But the fourth experiment that he did was really what told him what he wanted to see. What he did was he took that heat-killed S bacteria that was deadly, and he mixed it together with the living rough bacteria that was not deadly. And when that happened, the mouse died. Now, what that showed him was that there was some sort of what he called transforming principle. And this is the key to Griffith's experiment. If you're taking notes, Frederick Griffith, 1928, he did an experiment with mice where he discovered the existence of a transforming principle. Now, he didn't know what this was. He didn't know what molecule it was made out of, but it created the question that people then were able to try and solve, which is, what is that transforming principle? All right, so let's go to the next person. And this is actually a group of people, but Oswald Avery, okay, and a group of fellow scientists wanted to figure out what the transforming principle was in Frederick Griffith's experiment. And so what they did is they did a little bit more of a process of elimination. They used the same type of R strain and S strain bacteria. They used that same mixture that Griffith had injected here. And then they started to break it down to see what would happen if they were to destroy certain portions of that mixture. They were sure that the genetic material was either going to be protein, DNA, or RNA. At the time, we knew what all of these things were, but we didn't know what they actually did. And now a lot of people actually thought protein was the genetic material because it's by far the most complicated molecule. But Avery wanted to make sure. And so here's what he did. He took the solution that had been injected into that last mouse and killed it, and he treated it with 3 different types of enzymes. Each enzyme was designed to destroy a certain portion of that mixture. So first one was a protein destroying mixture. So all of the protein in this first test was gone, but we still saw living S bacteria show up. In other words, the mouse still would have died because the transformation still would have taken place. Then he treated another one with RNA destroying enzyme, and the transformation still happened. There was still S bacteria, that deadly bacteria that showed up. Finally, he treated a third one with an enzyme that destroyed DNA, and no transformation occurred. All of the bacteria was the rough strain, which is not lethal, and no transformation process happened. When we say transformation, what we're talking about is some information, some portion of the heat-killed S bacteria, that heat-killed deadly bacteria. And if you remember, alone, by itself, that heat-killed deadly bacteria was not harmful. But it was only when you combined the heat-killed bacteria with the living bacteria that the information inside of that deadly bacteria was able to somehow transform the living bacteria into something different. So, based on his experiment, Avery was like, hey, I figured it out. DNA is the genetic material. But the big problem is no one believed him. People assumed that because Avery's experiment didn't confirm what they had already thought, they just assumed it was wrong. So again, timeline's important. 1944, Avery presents his evidence, shows that DNA has to be the transforming principle, but the scientific community remained skeptical. They didn't believe him. So, further testing had to be done. That brings us to our third group. The third group is a group of scientists named Alfred Hershey and Martha Chase. And their experiment was basically designed to confirm that, sure enough, Avery's conclusion was correct, that DNA is in fact the genetic material and that it's not proteins. And so in this experiment, they set up basically 2 versions of it, pitting proteins against DNA directly. Now, the way they did it was they used bacteriophages, which is like a virus. And the way that a virus works is that it's pretty much made out of two parts. There is a protein coating on the outside. That's kind of the spaceship looking portion of it. And then there is DNA on the inside. So scientists at the time still weren't necessarily sure which part of the virus contained the genetic material. Is it the spaceship looking portion on the outside, or is it the DNA on the inside? And so what Hershey and Chase did is they radioactively marked both parts of the virus. They took one culture and they radioactively marked the outside, which is primarily made of sulfur. Well, primarily a protein, which contains a lot of sulfur. When they allowed this virus to attack a cell and begin to reproduce, none of the markers showed up in the reproduced virus. In other words, the portion that contained the genetic material did not pass into the cell because it wasn't reproduced. Now, the second one they did is they used phosphorus, and phosphorus is an important component in DNA. So they used radioactive phosphorus to mark the second group of culture. In this group of culture, as you can see, the DNA was injected containing the phosphorus material, and that is what actually reproduced. Hershey and Chase's experiment confirmed that DNA is in fact the genetic material that is passed on from parents to offspring. So again, quick recap. You need to make sure you're very familiar with these three scientists and what they did. First of all, Frederick Griffith worked with mice, discovered the transforming principle, discovered the idea that there is a transforming principle, but he didn't know what it was. Avery followed up on Griffith's experiment. He destroyed certain portions of that mixture until he ultimately concluded that DNA was the transforming principle, but the problem was nobody believed him. The scientific community remained skeptical. Finally, Hershey and Chase, they used viruses, they marked them with radioactive markers, and they were able to demonstrate that DNA is in fact the genetic material. And once this happened, scientific community accepted their results, accepted Avery's results, and then the job became, all right, well, now that we know DNA is the genetic material, we got to learn about it as much about it as we can. All right, so that's it for this one.